I'm Sonia. I work at the Tata Institute for Genetics and Society. I do a lot of work with Drosophila. I've done lots of clonal analysis. We're now working with mosquitoes, and we're trying to move some of these clonal analyses in there as well. So exciting times in general. So as you know, hello, why is this not moving? Yeah, okay. We're going to be talking about mosaics. We'll talk about what they are. We'll talk a little bit about how you can make them, and we'll try and see what mosaics or clonal analysis can tell you about biology in general. So I took the liberty of stealing this poster image, which I just found out, I was almost certain of it, but Deepthi has made. And the reason I picked it is because it just so beautifully summarizes what a mosaic is. So we're taught in biology in your most basic of classes that every single cell, whether you're talking about a cell on your skin or a neuron in your brain, those neurons or those skin cells look very different, yet their genetic makeup is exactly the same. So if you were to think of the fly in that term, this is what it would look like. Every single cell in that fly is exactly, the genome of every single cell is exactly the same as each other. But that's not always true, and that's why we have things called mosaics. So if you could imagine that some of those cells were actually genetically different from one another, then and you had that vision to be able to look in and see what the genome looked like, and you painted those cells according to those colors, then you'd get what Deepthi has drawn on the right? Each cell with a different genome. So that's what mosaics are. That you assume cells of an organism to have the same genome, but there are conditions, naturally occurring conditions, as well as those that we can experiment with, that make genomes within an organism, the cells in the genomes within organisms, heterogeneous. So I mentioned that you have naturally occurring mosaics. So since this is a class, can I sort of turn around and say, can you guys think of any naturally occurring mosaics? So organisms that have different genomic makeup within themselves. Is there anyone wants to make guesses at mosaics around them that they see? I'll start off with one that's not on my list over here. The most common one that you could imagine is the immune system, right? So all of your immune cells that make antibodies are genetically different from one another. That's how they can make antibodies against very specific proteins. They have this VJD recombination, resulting in slightly different genomes. So the immune system is one example. Can you think of others? Does anyone want to give it a shot? Naturally occurring mosaics? Okay, I'll give you one that is on my list and one that's an unfortunate example, cancers, right? So that's the problem with cancers, that you suddenly have a bunch of mutations which are genetically different, and so the cell, and often it's in oncogenic genes, these mutations, and so cells proliferate more than they should. More than that, even within cancers, the cancerous tissue will itself consist of heterogeneous genetic makeup. That's a property of cancers. Okay, anyone, come on. One.
C. elegans?
Okay, C. elegans. What about this?
They show sexual dimorphism. So since you have both the sexes, and then I'm assuming some cells are male and certain are not.
Excellent example. Okay, so I have something like that, gynandromorphs, not quite what you were talking about, but basically an organism which is both, normally not, so Drosophila is a picture of a Drosophila from a paper that's more than 100 years ago now. Drosophila have males and females, sexually dimorphic. But there are occasions where you will find an organism, a fly, which is both male and female on the two sides. So can you see my cursor? Can someone tell me if you can see my cursor?
Yes, we can.
Yeah, okay. So on this half of this fly is male, genetically male, whereas this half of the fly is genetically female. And you can tell that in this drawing because the male fly is smaller. Those of you who've worked with flies know this. You can see the sex comb over here on the front leg. And the abdominal coloration is slightly different. If you were to flip this fly around, then you would see in the genitalia, one half, this half, would be male, whereas the other half would be female. On this half, you can see the fly is larger. It doesn't have the sex combs. And if you flipped it around, it would have female genitalia on that half. Okay, so gynandromorphs.
May I ask a question here?
Yes, Sujata.
Yeah. Is this gynandromorphs only external morphology is like this, or even the internal is like that?
Wherever you have sexually dimorphic tissue, you will see it. You may not always see it, but if you were to look at in Drosophila, for example, where it's XX or XY, if you look at the male side, it'll be only a single X, no Y, but only a single X. And on all the tissue, all the cells on one side, if you were to probe for the chromosomes, because you can't always see the structures, right? But on the other half, it'll both be XX. All the cells will be XX.
Okay. Yeah.
So we'll talk about that in a second.
Sonia, is there a reason why this is half-half?
Yeah. Okay, 1 second. Just a second. So another example, just a second, Dinesh. Do I know you, Dinesh?
Do you know me?
I don't know. Deepti said that I should know you.
Sanjay's lab.
Oh, okay. That's Dinesh. Hi, Dinesh. Okay. No, so here's I'll come back to that in a second. So here's a final example. Sorry, I opened the window, but it's quite noisy. I'll shut it again. So here's a final example of a calico cat. So this is Nilofer over here. And if you'll notice, Nilofer has 3 colors on her body. She has white, she has black, and she has orange as well. And so this is another form of mosaicism. Now, all of these 3 examples occur because of different reasons. So cancers, of course, as you know, happen because of mutations and environmental cues, for example, chemicals or radiation could cause double-stranded breaks in your DNA. And when your cell is trying to repair it, somatic tissue we're talking about is trying to repair it, that repair doesn't occur correctly. And so you may get incorrect repair in oncogenic genes resulting in heterogeneous, genetically heterogeneous tissue. So that's cancers, one way in which you get genetic mosaics. Gynandomorphs, Dinesh. So this usually happens because of a ring X. So what happens is the X chromosome in rare and in an aberrant case forms loops at the end and forms what's called a ring X. Now this is a very unstable structure. So when the cell is actually dividing, what ends up happening is that that chromosome gets lost in one of the progeny. It doesn't get inherited equally. It doesn't get divided up and inherited equally in the 2 cells. Now, if this happens early enough, so imagine when the zygote is 1 cell and a ring X happens then when it's dividing into 2 cells, if it happens at that stage, then one of those cells has lost the X chromosome. And so now everything that comes out of that 1 cell, one side of the organism becomes male versus the other becomes female. You can get this loss can happen anywhere. And in those cases, you won't see a bilateral male-female difference. You'll see patches. But these are very dramatic to see. And actually, I encourage you to go look online at gynandomorphs. They're beautiful examples. Flies are not very sexually dimorphic, so it doesn't look as dramatic. But if you look at crustaceans, or if you look at animal butterflies, which have really dramatic sexual dimorphism, you see this really beautifully. Okay, and this example, I picked it because it's interesting because it doesn't actually happen because of the genome. It's an epigenetic phenomena where in many mammals you have something called dosage compensation. So females have 2 X chromosomes, male have only 1. You don't want the female to have lots of expression of things on the X chromosome. So one of those X's gets silenced. And this happens in a patchy way. And because coat color in cats is determined by genes on the X chromosome, you get this patchy kind of inactivation. So those are ways in which you can, and of course we've not covered plants. Plants are another set of organisms where you have beautiful examples of mosaics. You must have seen Barbara McClintock's corns, where each corn in the corn cob is a different color. And that's a totally different mechanism of getting mosaics. That's transposable elements jumping around and giving you different colors. So different ways in which you can generate genetic mosaics, and they show up quite beautifully in the world around us. Okay, so whenever there is such interesting biology out there, an experimental biologist will come and try and figure out a way of using it to come up with to answer a question that they are interested in. So just a quick historical perspective on how mosaics have been used and understood in biology. The first is, I don't know if any of you have done a developmental biology class yet. If you have, you must have heard of chimeras. So all early developmental biology was an effort of fate mapping. So they would take, people who were very interested in what parts of the tissue gave rise to what part of the embryo gave rise to what part of the organism. And the way they did this was they would take 2 embryos, very early embryos. They'd take one bit off one embryo and graft it onto another, and then watched to see what this became. Now this thing of making a graft is called a chimera. You end up with an embryo which is not quite one organism, it's a genetic mosaic. And actually chimeras are still used quite extensively. Avian chimeras are used between chicken quail, and amphibian ones are also used. So that's one way in which people used genetic mosaics to understand biology. Another actually, and we'll talk about this example a little more in detail, was in Thomas Hunt Morgan's lab. So again, going nearly 100 years ago, what Curt Stern noticed was, if you looked at the cuticle of the fly, he noticed that there were patches apposed to each other that looked phenotypically different. And that it's a beautiful paper. I urge you to go back and read it. He, just from looking at patterns of these twin spots of mosaics, he could deduce that mitotic recombination was happening, that recombination, not in the germ cells, which had been described in the lab before, they used that to do linkage mapping, but even in somatic cells, that recombination could occur. So that's twin spot Markham, twin spots. I'm getting ahead of myself. Okay, so a very important part of this, using mosaics in experimental biology, is the ability to see them. So for example, in the fate mapping, once you graft it, unless you have a way of looking at these, you will not know what this tissue is becoming, right? And so for example, yes, that's the example, that if you weren't able to label those cells, then they would be basically boring, like the silhouette of the fly on the left. But if you could actually see through the genome and mark those genomes that were different, then you would see what Deepthi has drawn over there. So in the case of the quail and the chick, this people do by staining these chimeras with a stain that labels heterochromatin. And now the interesting thing about this is that quail heterochromatin is nicely condensed and centered at the center of this nucleus over here. And the chick heterochromatin is much more dispersed. So right now you can look at this image and you can draw a line saying which part of this tissue is quail and which part is chick. So the ability to see the genetic mosaic. In the case of Curt Stern's twin spot, where he was able to deduce these very careful mitotic events that would lead to a yellow or a singe was his ability to see yellow or see singed phenotypes. And so that's just to give you an idea. So usually flies on their thorax have the nice long bristles and some short bristles. But in the singed phenotype, these bristles look like they're singed, like they've gone too close to a fire. So you can see those whirly, wiggly, instead of nice and long bristles over there. And of course, yellow you must be familiar with. Normally the flies have a brown body color, but yellow is very, very different. So in both of these examples that I've shown you, it's always the ability to be able to detect these genetically different cells within an organism that allows you to either use it or exploit it to understand some biology. Okay, so I said that we would talk about this, the twin spot mosaic of Curt Stearns, a little bit more, because really that's the one observation that has launched all of this ability to do mosaic analysis in flies and other organisms. But before we do that, I didn't know what range of people with what background were here, physicists and whatnot. So I thought we'd do a quick recap of mitosis and meiosis and recombination, because all that really lies at the heart of being able to generate clones. So I've just inserted, I've been lazy, I've inserted these videos. Well, not just lazy, they do a much better job than I would. So I will play this. I won't play the whole thing, but just up to a certain point that's relevant. Please let me know if you can hear the sound.
During the mitotic phase, a cell will undergo mitosis to form two new nuclei, and then divide to form two new individual cells during cytokinesis. Mitosis is the process of dividing the duplicated DNA of a cell into two new nuclei. Mitosis is split into distinct stages. The first stage is prophase. The DNA condenses, organizes, and the classic chromosome structure appears. Next comes prometaphase, where microtubules attach to the chromosomes. This step is followed by metaphase, where the chromosomes align. Metaphase is followed by anaphase, where the chromosomes separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes.
Okay, I'm going to pause it there. Can everyone tell me if they heard it? Could you hear it?
Yes, ma'am.
Oh,
yes,
please call me Sonia. Anurag, please call me Sonia. I'm going to take that as yes from you. Okay, so remember that's mitosis. And we're talking about mitosis because we're talking about somatic. All this entire lecture is about somatic mosaics, somatic recombination, not germline. Germline does totally different things, and we're not going to talk about it except in this one. Oops, I'm sorry. Oops. Except in this next slide, where I basically want to highlight the differences between mitosis and meiosis from the perspective of recombination. So I'll play this. This is slightly longer, but I think it'll clarify a few things. So please bear with me.
In biology, there are often vocabulary terms that sound pretty similar. Chromosome, chromatid, chromatin, transcription, translation, mitosis, meiosis. You probably have encountered this. When I was first learning about mitosis and meiosis, I learned them both separately first. And then I tried to figure out what was the same about them, what was different, why did they both matter? I would try to compare the stages by flipping through images. You know what would have really helped me? A side-by-side comparison. And that's what this video is. We assume you already have a background of mitosis and meiosis. If not, take a look at our videos on them. But this video is a side-by-side comparison, presented in a split screen. Mitosis on the left, meiosis on the right. Both of these processes, along with the cytokinesis that follows them to split the cytoplasm, are involved in making new cells. Mitosis results in body cells. Meiosis results in sperm and egg cells. otherwise known as the fancy term, gametes. Before we start mitosis and meiosis, let's look at what you start with. Your starting cell in both mitosis and meiosis is diploid. Written here is 2N. That means it has 2 sets of chromosomes. In humans, that's including 1 set of 23 chromosomes from mom and 1 set of 23 chromosomes from dad. 46 chromosomes total. During interphase, the cell duplicates the chromosomes. When you duplicate 46 chromosomes, you still say there are 46 chromosomes if the newly duplicated portion is still attached at the centromere region. But there are actually 92 chromatids. Interphase isn't part of mitosis or meiosis, but it's a really important phase because it duplicates chromosomes before we get started. Just to point out, it's really hard to draw 46 chromosomes. We're going to use 6 chromosomes in our diagrams when we illustrate what's happening, because it's much easier to draw and visualize. Oh, and just a fun fact, some insects have 6 chromosomes, like mosquitoes. Unfortunately, I'm not a fan of mosquitoes, but mosquitoes do both mitosis and meiosis. When learning the stages, we get the acronym PMAT, which is helpful for understanding the stages. Both mitosis and meiosis go through these stages, but meiosis goes through them twice, and therefore has a number next to each PMAT stage. We're going to show some basic events for each PMAT stage, but please know that there is way more detail to explore than what we can include in this quick video. Prophase in mitosis. Remember that pro can mean before, and this stage comes before the others. The chromosomes are visible if we say they're condensing, which means they are thickening. Prophase 1 in meiosis. Happening here too, but the chromosomes are actually going to match up with their homologous pairs.
So this is very important.
Means that the chromosomes are approximately the same size and they contain the same types of genes in the same locations. With each pair, one came from mom and one came from dad. In this formation, chromosomes can transfer the genetic information and exchange it between each other. It's called crossing over. It can make for what we call recombinant chromosomes. Metaphase in mitosis. The nuclear envelope which had surrounded the nucleus was already disassembled before metaphase even started. For metaphase, I like to remember the M for middle, because in this stage, the chromosomes line up in the middle of the cell in a single file line. Metaphase 1 in meiosis. The chromosomes are in the middle as well, but they're still going to be in pairs in the middle of the cell, so it's not a single file line. Anaphase in mitosis. I like to think of the A for away, the chromatids are...
Okay, I'm going to pause that there because I think that covers what we need to discuss. So notice how in this discussion of mitosis and meiosis, in meiosis we talk about homologous recombination because it's true, there's a lot of homologous recombination during meiosis. And it happens at that time of metaphase 1, where the homologous chromosomes come together and there can be recombination. That doesn't occur in the metaphase 1 of mitosis. The homologous chromosomes are not aligning up, so that opportunity to do recombination doesn't occur. But that's not to say that recombination doesn't occur in somatic cells. It actually does. Oops, that's not what I meant to do.
Are pulled away by the work of the spindles. They are moving.
Okay, recombination does occur in somatic cells. It happens before mitosis. So during interphase, when the chromosomes have duplicated their chromatids, that's the stage at which the homologous chromosomes can actually come together and have recombination. It's very rare, but it does occur. And then remember, once after that, everything is as in this video, where you have the chromosomes lining up in the metaphase, and at anaphase they get pulled apart. Do people want to discuss this, or is this clear enough? And maybe if there are doubts, we can come back to it. Or Sujata, were you going to say something?
Can I ask a doubt? I mean, if you can decide if you want to answer it later on also.
Okay.
My question is that what is the reason for this rare events of recombination in mitosis?
Why do things occur is a tough question to answer. Sometimes it happens because there's there's a double-stranded break. And the cell will just try and repair any break. And so when it's trying to repair a break, sometimes it uses a homologous chromosome. So if it happens to encounter, and it's in the milieu, it happens to encounter the homologous chromosome, it'll repair, and by accident a recombination will occur. Yeah, I mean, I guess that would be my answer, that mistakes do happen, double-stranded breaks do occur, and in a process of trying to repair it, a recombination event may occur. So the other thing that actually, even from the time, from nearly 100 years ago, what people realized was that somatic recombination rate increases upon irradiation. So this was Müller's work, and he actually won a Nobel for it in Drosophila. He showed that you increase your irradiation of these organisms, and you can get much higher recombination frequencies, both somatic and germline.
Okay, and one more related question, because recombination is a fairly complex process, right? You have to exchange the parts of chromosomes, so you will need a lot of supporting enzymes and macromolecules. So all those should be present in the cell which is undergoing mitosis.
I'm sure they will be, because it's so important for a cell to be sure that it doesn't create breaks. It repairs anything as quickly as possible. Okay. So definitely at all times, I think that's a whole different field where people study actually how a break in the DNA can recruit all of the machinery required to repair that break. I'm not familiar with that, but yes, this is it's very true. The cell is always ready to repair DNA breaks because it's so dangerous for it, right?
Yeah, right. Yeah. Thanks.
Okay, so basically, so that's the background with which we'll go into this. And if there's a doubt about any of this as we start talking about the methods, please do stop and we can discuss them. Okay, I don't want to play the movie again. Okay, so I said that we would talk a little bit about Curt Stern's twin spot experiment observation that has basically launched clonal analysis in flies and in other organisms as well. So remember that I told you that he looked at two phenotypes, yellow and singed, yellow body color and singed hair, bristle on the back. Now, so we're gonna step back and see. I mean, he just looked at the flies and deduced what was happening in the cell. We're now gonna go into the cell, see what's happening, and then see how it resulted in the things that Curt Stern saw. So here's a cell. Imagine this is a cell on the developing wing disc, which would give rise to the cuticle of the fly. And this cell, like every other cell in this developing larva, is heterozygous for two loci. Yellow, it's heterozygous for yellow. So it has one mutant allele of yellow and one is wild type. And it's heterozygous for singed. So that is one copy of singed is mutant, the other is wild type. Meaning these are both recessive alleles, meaning that if nothing were to happen, if everything was normal, then this fly, when it came out, would look perfectly normal. It would be brown body color and it would have perfectly formed bristles. But in these rare events, imagine that there's this one cell that something weird has happened. Well, first of all, in this cell, when it's preparing for mitosis, it will duplicate its chromosome, and now you have two chromatids per chromosome. And as a result, yellow will get duplicated, singed will get duplicated. And now that rare event that we were talking about, imagine that something happens and there's a lot of irradiation around, and then a recombination event happens right next to the centromere. Now, because that event is happening right next to the centromere, what ends up happening is that entire chromatid gets swapped. Is that clear? So if a recombination event happens here, this whole chromatid will get swapped onto this, and this whole chromatid until that point of break will get swapped onto that. And what you'd end up with is this. Is everyone with me on that? Okay, I'm going to take that as a yes. Now imagine we're going to metaphase. So now in metaphase, everything is lining up. These two chromatids are lining up at the at the equatorial plate. Is that what it's called? I'm not sure. Metaphase plate, something. And there are a few things that can happen. We'll consider the example that we're interested in. Okay, so in this event, now those microtubules that have hooked on to this centromere are going to pull these two chromatids apart, and what you're going to end up with are two cells from that one parent cell, two cells where one of those cells is mutant for both the loci for yellow are mutant now, and in the other daughter cell, both the loci for singed are mutant now. Is this making sense to people?
Yes.
Yes. I'd like to hear a no. Is any? Please do say if it isn't making sense. We can go over it again, because this lies at the heart of all of our.
What's not clear to me is how this recombination happened. Is it or maybe I'm jumping ahead of myself.
No. It's so. This is again we were talking. This is what Sujata was saying. It's a rare event, Dinesh. Most of the time, if this was just growing in the lab, you would not have seen anything out of this talk. You just keep growing it, and it will be totally normal. One in an X number of flies will have this. Now you can increase the frequency of that by increasing the incidences of double-stranded breaks. And so they did this. So they irradiated using Muller's method of just zapping it with irradiation. They increased the frequency of making these double-stranded breaks and relying on repair. Now there's so much more to this. If you go back to Curt Stern's paper, actually, I very conveniently showed you a recombination event that happened over here. But that double-stranded break could occur anywhere along all of this. And so in his paper, actually, he has these beautiful tables of what proportions he saw of things, and therefore went back and worked out where the recombination event could be occurring. Because you can imagine if the recombination event occurred here, you will not have this outcome. You will get, if it happened over here, I guess you would end up with normally wild type and in occasion singed patches. You wouldn't see this twin spot. But it's the twin spot that people now exploit in experimental clonal analysis, which is why we're focusing on that. But basically, you're making double-stranded breaks. You can increase the frequency of increasing double-stranded breaks by irradiating. Or people then realize chemicals, EMS then became a common way of, but that's not double-stranded breaks. But basically, many environmental influences will create those breaks, and you can. Initial clonal analysis relied on that, but we don't anymore. We got smarter, and we'll talk about that.
Sonia?
Yes?
Can I ask a question?
Yes, Sujata.
Maybe it's a stupid question, but just to make sure that I'm understanding the things correctly. For this kind of recombination to occur, you need the double-stranded break occurring at the same level in the two chromosomes, right? If they occur at different levels, then this cannot happen. Am I right?
It's a double-stranded DNA break, and so it'll use even if it happens in one, it's going to use the homolog, and then when that machinery so when DNA repair happens, when there's a double-stranded DNA break, it's going to find its partner, and it brings its partner in, and when the repair machine, this doesn't have to be broken. The repair machinery will read off this, and that can create. Sometimes that relies on another break here to be able to repair. So, so it's the matching up of the homolog that results in the recombination event.
Fine. Yeah.
Okay. Okay. So what this would then result in is Curt Stern finding on that rare occasion that on the thorax of the fly, one patch of cells which were yellow-bodied in an otherwise brown background. These hairs will be wild type, whereas this other patch sitting right next to it had wild type cuticle color, but these singed bristles. And this is what is the twin spot. Okay, so again, yeah, just to reiterate that this observation of Curt's turns is what has launched the entire toolkits of clonal analysis that we use today. Okay, so let's pause over here and maybe ask you guys that now that we know what mosaics are, can you think of how they could be used in experimental biology? We'll keep revisiting this question, but I thought if this has jogged any ideas about how you could, if you were in the 1936s, in 1937 you read this paper because it took time for you to get a copy of it, what would it trigger in your head saying, aha, I could use it for this? Are there things that come to your mind?
I remember the question I asked Deepthi, and
Okay.
she sort of said that you will talk about it. I can see why. Probably labeling individual neurons. That's not 1937,
Absolutely.
but now.
Yeah, but I don't know how you're getting to that from Curt Stern's experiment, but yeah. Okay, labeling neurons. I'm going to write that down over here. I don't know if you guys can see this, but okay. Anyone else? Oh, right. I've shifted the. Okay, no ideas so far. Okay, doesn't matter. Let's go ahead and we'll come back to this question. Okay, so of course, now we're not going to rely on these rare events of double-stranded breaks and repair happening to get clones. What we'd like to do is to speed them up, and we don't want to be using X-ray radiation in the lab to speed things up. We want better, more clever ways of making clones. So, so what is it that you need to do to be able to get these more efficient ways of getting clones? The first is something we've already referred to. You want improved ways of seeing these clones. So that entire field has evolved. People from cuticle color and bristle shape, you don't have cuticle color and bristle shape in the brain to be able to see neurons. So that won't work. But people then started using eye color, again, won't work in the brain. LAC-Z, which is an enzyme from yeast, which causes where in the presence of a substrate, gives it color, which you can observe in any tissue that you express this in. So that was an improvement. And then, of course, came the fluorescent proteins, GFP, RFP, and variants of GFP and RFP. And then, of course, small tags, because now you can have very many different types of tags. So there's flag, there's V5, there's OLAS, there's HA, a whole bunch of tags. So new and improved ways of visualizing the clones in diverse tissues. It could be viscera, it could be cuticle, it could be eye, it could be anything. The other is increased efficiency of generating these clones, these double-stranded breaks. And there are very many tools that have come up that allow you to do this. So some of them I've listed over here. The flip FRT system, this is from yeast. This is what we'll talk about the most because it's the most used in clonal analysis. There's in flies, there's the Cre-Lox system, which was adopted from viruses. That's used much more in vertebrates. Cre-Lox turned out to be toxic for flies. So it is used, but very rarely. The ATP, you may have heard about this from Sonal. I found out that Sonal's already given her talk. And then there's KDRT, which I don't know which bug that's from, maybe Neospora or something like that. I don't know where that's from, but that's another system, but that very, very few people use, so I won't talk about that. Primarily, we'll talk about the flip FRT system, which allows you to increase the frequency of these recombination events that are otherwise so rare. Okay, so the
Sonia,
flip, yes.
What do you mean by clones here?
So you know those mosaic patches that we were talking about? So, so the organism, so take, for example, Curt Stern's observation. So the animal was, the whole animal otherwise was a wild type body color, brown, and the bristles were long and nicely shaped. In that, there were these patches of cells, which were yellow mutant, and patches of cells, which were singed mutant. These patches are called clones because they arose from one event. So one cell from that, remember we have that example where that one cell, the recombination event occurred, it resulted in 2 daughter cells. Now each one of those daughter cells, that one cell was yellow mutant, that one cell was singed mutant. That one cell divided, but because its genetic makeup is now different, all of its progeny were yellow mutant, all of this one's progeny was singed mutant. And so that entire patch of yellow mutant cells is called a clone. So it's a clone of that one original cell. And this entire patch of singed mutant cell is singed mutant, that entire patch, because and it's called a clone because it arose from a single singed mutant cell. Does that make sense?
Yeah.
Okay.
Sometimes we see this in the eye also, right?
Yes, absolutely. So that's a beautiful example, Shweta. So if you've been making clones, or I don't know how else you would see it. If you have a fly stock which has an eyeless flip in the background and some p element insertion with a W plus hanging around somewhere, you will see patches of eye color which are W plus and W minus. That's again a clone. Yeah. And actually that was used by Utpal Banerjee's lab with a bunch of undergraduate students to screen for genes that could affect eye development. So that's how they screened for it, looking at eye color mutations. Clone sizes clones in the eye using eye color. Okay, so FLIP-FRT, it comes from yeast, and it's basically, so FLIPase is an enzyme, and the enzyme recognizes these DNA sequences that are called FRT, FLIPase recombination target sequences. So it's just a string of DNA sequences. And it consists of a palindrome and a spacer in the middle, and you can have strings of these, and when you have 2 FRT sites, the FLIPase can bring those together. And when it brings those together, it can cause a recombination event. Now, what the outcome of that recombination event is depends on the orientation of the 2 FRT sites. So some of these events can result in an excision of any intervening sequence of DNA. So for example, you can imagine here are 2 FRT sites. I've just indicated them as arrows. They're just sequences of DNA, and they're oriented in the same orientation. And when they're in that orientation, any intervening DNA sequence, which I've put as a green, magenta box over there, it's just a DNA sequence. If FLIPase comes and acts on these 2, this gets excised away, and you end up with just the FRT sites. Now, how that occurs is basically there's a loop that happens. The 2 FRT sites line up. The recombination occur. I couldn't do animations like this or drawings good enough, but basically this purple green region would loop out over there, recombination would occur, and this would excise away from the DNA. Repair would happen over there. Does that make sense? There's a better picture later on, so you could see. So bottom line, if the 2 FRT sites are oriented in the same direction, then FLIPase acting on those sites results in an excision event. But an inversion can occur, and this occurs when the 2 FRT sites are in opposite orientation to each other. And this results in a loop that allows that DNA sequence to flip over. And so I've indicated that as, the magenta and the green have flipped over. So excision in tandem, inversion when facing each other. And then there's a final one, which is translocation, where if you put the 2 FRT sites on, say, 2 homologous chromosomes at the same location, then, and that's what I've indicated over here. So imagine the centromere is somewhere over here. Then the recombination event will occur at these sites when FLIPase comes in. And what you end up with is swapping those chromosome arms. So can you see how now we we can use this translocation to bring about the twin spots that Curt observed just by placing FRTs at those sites, instead of relying on a random event of DNA breakage.
So Sonia,
Yes, Ankit?
in translocation, are you flipping the entire arm or just a part of DNA?
Anything downstream of that FRT site would get swapped. So if the DNA sequence ended over here, this is all that would get swapped. But if this DNA sequence went on and there were 10 other genes down there, and this sequence went on and there were 10 other genes, all of those would get swapped. So it's basically just exactly like your double-stranded break, but you're engineering it through flip FRT. It's just swapping that chromatid, like we saw in the Curt example.
So
thank you.
Just a clarification and continuation. If that's true, in the inversion case, I'm guessing the color of the right one should not change, right? I mean, the first one should still be dark blue and the second one should be light blue.
So that's, I'm drawing it like that, but actually you get halves. It's a half thing.
Oh,
so the two palindromes, so each FRT is two palindromes. So these will get flopped. So if I were to, I would have to draw them as two different, each FRT is two different colors, and they would get inverted. So if this was dark, okay, if this was blue and, okay, can you see if I draw here? If I draw here, no, right? Okay, wait. Can I draw here? Okay, wait, let me try to explain like this. So basically there's a palindrome, right? So there's a palindrome this way and that way. And there's a palindrome this way and that way. The inversion will happen across here. So half of this FRT and half of this FRT is what would get swapped. So I've approximated it like that, but in effect, it's halfway through it gets swapped.
Makes sense.
Okay, any more questions? Okay, I'm gonna go ahead then. Okay, so much better drawings. How did people use the flip FRT system in biology? So here is an example of translocation. It's exactly like the Curt example. So imagine now, instead of Curt to Crow, so here's the centromere, and here are engineered FRT sites right next to the centromere. And instead of singed and yellow, we have RFP over here and GFP over here. And now imagine exactly the same things that we discussed a few slides ago. This would duplicate. A recombination flipase would come in, and a recombination event would occur, because now it will occur at that site now, because flipase will recognize that FRT site and cause the recombination. And now they haven't shown that intermediate step, but you would have green come up over here and red come down over here. Your Ankit, see everything downstream of that FRT site would get swapped. And then when metaphase happens and independent assortment occurs, you would have cases where this chromatid would swap with this, resulting in a cell that had two RFPs. And this cell would inherit two GFPs, resulting in the twin spot closely abutting RFP and GFP cells instead of singed and yellow. Does that make sense? How you can use flip FRT, a translocation usage of flip FRT to generate twin spots. Is that making sense? Is that a little cat? A dog? Is that making sense, guys?
Yeah, got it.
Yes, yeah.
Okay, great. So that's how you use translocations and flip FRT to make clones. But you can use excisions as well. And what happens in an excision? So here's an example. You have those two FRTs in tandem. Remember, they have to be facing in the same direction. And sitting in the middle of that is say, a ubiquitous promoter that's driving RFP expression. Now, here's the much better example. So what happens is it'll loop over onto itself to align those two FRTs together. Now, when the flipase comes in, a recombination event will occur, leading to the excision of this, which means that you've gotten rid of the ubiquitous RFP. And what that results in is a patch of cells which is not RFP in an otherwise background of RFP. So Dinesh, this patch of no RFP would be called a clone. And those two red and green spots would be called clones. So is that clear how you can use excision to flip FRT mediated excision to make clones as well?
Yes.
Okay, so now the wonderful thing about this is flip can be conditional. The enzyme can be under your control. What do I mean by this? So one is, we kept saying now you come in with the flip. I use that phrase, you come in with the flip. This is the wonderful thing about experimental biology, right? So what can you do? You can take the flipase protein and you can hook it onto a heat shock promoter. Now that means that in this genetic fly, if you raise the temperature to whatever, say 37 degrees for 15 minutes, the flipase gene gets expressed and whatever the rest of it will happen depending on what your genetic background is. So that means it's in your control. You can raise the temperature whenever you want. So that's really convenient. You can get, because we're working with flies, other organisms as well, but particularly because we're working with flies, you can get tissue-specific promoters. So I think Sujata was talking about the eyeless, the clones in the eyes, right? So that you can get by the screen that I was talking about with Palbanerjee with the undergrad students, where they looked at what are the genes involved in eye development. They actually used a flipase which was hooked onto a promoter that's expressed only in the eye, the eyeless flip. So you get tissue-specific promoters. You get UAS flips. So this is another way of getting tissue specificity in flies, where you have GAL4 UAS. I'm sure you've heard of that because you're at the end of your course now. So then you have UAS flips. Now if you have any tissue-specific GAL4, you can get tissue-specific expression of flipase, and all of these events would happen in that tissue. And then finally, there's this wonderful library of enhancer trap lines with flips in them. So basically you can then screen these. So they've gone and sat somewhere in the genome. That means some random enhancer, which you may or may not know what it is, will drive flipase expression in some tissue. And now you can screen it for a tissue that you're interested in and use the flip from there. So basically, the FRT is how they're oriented, give you all of these events, but because the flip is under your control in all of these different ways, you can get much more sophisticated control of the system. Okay, so now what can
I have a doubt.
Yes.
So when we use UAS flip, so we can also achieve a temporal control by coupling it with GAL18. Right?
Absolutely, yeah, exactly. So this is the bottom line. All of this is to just throw the tools out there at you guys, and you guys have to think of how you want to use this for your experiment, because you'll see as we go along that there's so many different ways in which people use it. Okay, so twin spot clones. So how have people used this twin spot? I've just told you a bunch of techniques. I thought it would be nice to see how people have used it. So the first example of using this kind of flip FRT mediated twin spot, I like this example from Laura Johnston, where she's actually looking at growth. Like what controls the rate of growth of cells. And here's a wing imaginal disc, where she's got some cells which are borderline, some low level of GFP. She's got some cells which have no GFP at all, they're clean. And she's got some cells that have really high GFP expression. And basically the genetic background of this is this. So it's exactly the setup that you're familiar with by now, where you have the centromere, you have the two FRT sites. We've now put in FRT sites right next to the centromere. Downstream of one homolog, on one chromosome, is a mutation in a gene called MYC. And on the other chromosome, the homolog, you have GFP. So this is exactly the same scenario as Curt's turn. It's heterozygous for MYC, a recessive allele, and it's heterozygous for this thing that you put in, a ubiquitous promoter with GFP. So now all of these cells over here, which are low-level GFP, are probably this genotype. Heterozygous for the GFP, heterozygous for the recessive mutation. But then she would have come in with the heat shock, and she would have given, like, say, 15-minute heat shock at some early developmental time point, L1, say. And what she observes then are these, and she dissects it at L3, and she observes that some cells have no GFP. And it's likely that those cells, because of all of that recombination event, have resulted in become recessive, have inherited both the mutant copies of the MYC gene. So they're now homozygous null for MYC, but also notice they've lost GFP. That's why there's no GFP in that patch of cells. But the twin spot, so that's these cells over here, the much brighter color, would be the twin spot of this event, meaning that they would have inherited both copies of the ubiquitous GFP. And so that's why they're much brighter GFP than the background. And what she noticed in her experiments was that the cells that were MYC null were very small compared, so that's the clone, compared to the twin spot, which is much larger. The wild type, which is 2 copies of GFP and both healthy copies of MYC, are much larger than the twin spot. You'll see it over here as well. The MYC null is much smaller than the twin spot, which is much larger. And then you can do the converse experiment. So this is so basically what this is saying is, upon loss of MYC, you have some sort of a retardation of either cell division or growth, something like that. But you can do the converse experiment, which they did, which was to say what happens when you misexpress. Based on this, you would expect that the clone sizes should be larger. So that is what she sees. So when you have a GFP control, wild-type clones are small. But when you misexpress GFP in those clones are much larger. So exactly as you would predict. Now, the interesting thing about this is how we did this. So this we discussed is effectively, we've discussed this, but in this experiment, she used a different approach to make the clone, which is, okay, so we're now moving from pictures like this to genetic annotation. So whenever you see, it's used differently, but usually if you see something like this, these, more than arrows, they indicate FRT sites. Sometimes they can indicate other things, but depending on context, you'll know. But so these are 2 FRT sites in the same orientation. That's what it's trying to indicate. And within that is sitting this CD2 sequence. And that CD2 sequence, you can't see it in this genotype, has a stop cassette. Now, on one side of that FRT is the actin 5C promoter, so a promoter that should be expressed in all cells. But that promoter can do nothing because of this CD2 and stop cassette, even though there's a GAL4 UAS GFP over there. So the promoter is unable to drive GAL4 UAS GFP because of this stop cassette in the middle. And that would be all your dark patches over here. Okay, but now you can come in with your heat shock flip. And what will happen is, because anyone wants to guess?
exclusion?
Yeah, so because you have your two FRT sites in tandem, you'll have an excision of that CD2, and now actin can drive GFP. So that's your two positively labeled clones. Now remember, because this is now actin driving GAL4 UAS GFP, you can put in a UAS DMIC, which is what she has done over here, which is what she's indicated. So in this one picture, see how the two methods of using flip FRT has given you different ways of analyzing a question. So the translocation scenario allowed you to make homozygous mutant clones. You can't do that with this excision event, which allowed you to misexpress a gene. You can't use the excision event in this setup to create a mutant, and you can't use the translocation to create an excision. But you can use, depending on how you orient these things, you can do overexpression or mutant analysis. Okay, so of course, the problem with this whole thing is, unlike the schematic I showed you, the mutant cells that you're interested in are actually negatively labeled. You can't see those clones. So actually, there was another piece of work which I like very much, but didn't somehow get used so much in answering mechanistic questions, was a twin spot generator, where actually you could label the two different clonal patches in different colors. But we won't talk about that. I just like that they were able to develop that. Okay, so the problem that we discussed was that in the twin spot that the Laura Johnston paper I showed you, the mutant, you can't see. You know that it's mutant because of loss of label. But ideally, you want to be able to see the mutant cell. What happens to that cell when it's lost this gene that you're interested in? So you want to positively label a mutant clone. And of course, you want a clean background. You don't want this heterozygous GFP, which is low-level GFP, and then you're looking for differences in higher level or total loss. So you want a clean background, and you want a positively labeled mutant clone. So here enters the I don't know, godfather of clonal analysis, which is mosaic analysis. We've talked about this with a repressible cell marker. That's the key, repressible cell marker, and its acronym is Markham. So this really transformed how people did clonal analysis. So let's get into, yeah. So it uses, in terms of tricks that it uses the GAL4 UAS system. It uses the flip FRT system. We've discussed this. Also uses that repressible cell marker, which is GAL80, which I think Shweta spoke about. So it uses a combination of these things to achieve positive labeling of a mutant clone against a clean background. So let's talk a little bit about how they do this. And this was developed by Shuwen Lee and Lichun Luo in 1999. So, okay, it's a bit involved in the genetics. So I'm going to go through it step by step. This figure is not from the original paper. Yeah, it's from this, but I think it explains it better. So I used it. So let's go through it step by step. Look here at this GAL4 line. So this is a tubulin driving GAL4. You can imagine any GAL4, but this is just a generic one. It will be expressed in all cells. Now that GAL4 in the same genetic, so remember every single cell in the fly has this. So in that same cell, there's also, there's also UAS GFP. So ideally this cell and every cell in the fly should be expressing GFP because it's tubulin GAL4, right? But it doesn't, and it doesn't because there is this repressible cell marker that Markham is famous for. So you'll notice the GAL80 that we refer. Is everyone familiar with GAL80? Have you guys covered GAL80? Okay, I see Dinesh nodding, so I'm going to assume it's been covered. So now there's tubulin, the same generic promoter driving GAL80. So now what's going to happen? Now that what we had earlier inferred that the tubulin GAL4 will drive UAS GFP, every single cell should be GFP positive. It won't because there's tubulin GAL80 there, right? But now I want you to pay attention to the third element, which is the FRT sites. So this fly, tubulin gal4, UAS GFP, tubulin gal80, also has FRT sites, and those FRT sites are interestingly located. So if you look at this chromosome, now this is again the same configuration that we've seen so far. Imagine the centromere is right over here, FRT sites right over there. That tubulin gal80 is actually downstream of the FRT site. Okay, and if you look at the homologous chromosome, there is your mutation. Imagine Mick. So Mick is sitting over there. So now what happens? Now you come in with your heat shock flip. Come in with your heat shock flip. You raise the temperature. Flipase gets made, these hexagons over here. It acts on this and a recombination event occurs. Now imagine all of that scenario of metaphase plate independent assortment, and there are two likely outcomes of this. In one case, what you'd end up with is a cell that inherits both the gal80s, and the converse of that event is that the other cell will inherit both the mutations, the MYC mutations. Now, as a consequence, what you'll notice is that both the gal80s have gone to this cell, leaving this cell free of gal80. So can you imagine what would be happening to this cell? Any guesses?
Expressing GFP.
Exactly, it's lost gal80, it's expressing GFP. Not only that, it's homozygous mutant for MYC, right? So you've positively labeled all the background cells don't have GFP. Your mutant cell is the one that has GFP. Is that making sense, guys?
Yes.
Okay, great. So what this amounts to is this. So let's focus on C over here. So these are your GFP labeled cells. And because they're GFP labeled, the logic, the genetics tells you that it should be MYC mutant. And in red, actually, they've stained for MYC. And you look, if this is the clone, the corresponding patch over there doesn't have MYC. You can see that over here. This is the neighboring clone, null for MYC, and that's the merge over there. So Markham allows you to positively label a mutant clone in an otherwise clean background, clean and otherwise heterozygous background. Okay, that was an example of the wing disc. But what Markham has done is really transform things for nervous systems, how we analyze nervous systems. And so in the same paper by Lee and Luo, they had their next figure actually was to do with the nervous system. So what you'll see in B is a bunch of, say, 100 cells. I'm just throwing a number out there. And the tract in the larval brain. What you see in C are 2 cells labeled with the tract labeled as well. And what you see in D is a single cell with the entire tract labeled. What this allowed them to do, and they had hints about this from very early experiments where people had actually gone in and put dyes in cells in the developing embryo and shown that when a neuroblast divides, you have this pattern of division. So this was kind of known. So you have one neuroblast which divides. Neuroblast is the stem cell that makes neurons, and each neuronal stem cell, neuroblast, divides asymmetrically. So it self-renews into another neuroblast, and it makes this cell called a ganglion mother cell. The ganglion mother cell now divides once and once only to make 2 neurons. So what they deduced from this Markham experiment was that in events such as B, they didn't see just one of each event. They saw these categories of clones where there were many cells labeled, 2 cells labeled, or 1 cell labeled. And they were able to deduce that if you have many cells labeled, the loss of gal80 is likely occurring in the stem cell, which is why the rest of that lineage, all the cells that are born from it, will not have gality and therefore will be positively labeled. Does that make sense? So if gality is lost in the neural stem cell, all of the progeny would have lost gality, and therefore all of the progeny of that one stem cell will be labeled. Now, if in this scenario, what likely occurs is that the gality gets lost at the time of the GMC forming. Now, if the GMC loses the ganglion mother cell loses its gality, then its two progeny will have lost gality and therefore will express GFP and not the entire lineage, right? That's what you see over here. And then finally, it's also possible that in this last division of the GMC, one of the cells loses its gality, and so you positively label only one cell, which is what you see. So whenever they did these experiments, they found these three categories: many cells, two cells, one cell, neuroblast clone, GMC clone, single neuron clone. And this actually was transformative in trying to understand how the brain is built in flies. So this was the first paper that did this, but it's been used by many, many, many people afterwards. So here, basically, they used exactly that type of Markham analysis in the antennal lobe, one of the stem cells that make projection neurons in the antennal lobe. And what they found was that, remember, this is, you come in with the flip, right? You have heat shock control over the flip. So these authors, they gave heat shocks at different time, 20 hours after larval hatching, 40 hours after larval hatching, 60 hours, they kept giving heat shocks at later and later time points. And they kept focusing only on the cells, on the single cell clones. So what they could do was that if any single cell clone was labeled at this time point, it was likely born after the time of the heat shock. Right? Am I making sense, guys? So only if, let's go back here. So if you give a heat shock here, then this cell will never be labeled, right? Because it can never lose gality. It's already divided, it's already formed. You need that cell division, right? Metaphase and dividing for it to lose gality. It will not, this neuron will not lose gality unless it was dividing at the time of heat shock. Am I making sense? Please tell me if I'm not. Yes, okay, yes. So in that way, they used Markham and, serial heat shocks to make clones and back calculated exactly which neuron was born at what time. So it's very, very powerful to do lineage analysis, birth dating of neurons within a lineage. Okay, yes.
So I have a question. Maybe I'm not able to see the time. How long can you give this heat shock, and how many how many division events can happen? To what level can you do, to a point where it is understood that beyond this, the clones may not survive?
Okay, you're asking. So the clone surviving is not a matter of how long you give the heat shock. The animal surviving is usually what the problem is. And ideally, actually, you don't want to give too long heat shocks. You want to refine it such that you have small events. The longer your heat shock, what will happen is anything dividing, the recombination event will occur in anything that's dividing. So if you extend your period of heat shock, so suppose you give it for, say, 1 hour, you may catch, say, 12 cells that were dividing. If you give the heat shock for 15 minutes, you may catch only 2 cells that are dividing. If you give the heat shock for 5 hours, you may catch 50 cells that are dividing. So this 5-hour event will end up with very many clones in the brain. The 15-minute heat shock will give you very few clones in the brain. So you have to kind of tailor it to what your requirement is. In this experiment, you want very refined because you're trying to birth date neurons. So you don't want to give too broad a heat shock because then you won't know when in this 5-hour period this neuron was born. You want to give refined heat shocks. So I believe for this experiment, they were also using a heat shock clippase, which was more messy and perdured for longer, but they used 1-hour heat shocks. Nowadays, people use more like 50. I mean, it depends on usage, but the clippase is much better now and more efficient. So you can make do with, and then it gets, they've hooked it onto these pest domains, so it gets rapidly degraded. So you know that whatever your events you're seeing was occurring within that 15 minutes, and maybe a little bit more than that, because by the time cells divide, maybe say a couple of hours window.
So this also is a little bit dependent on the tissue type as well, I guess.
Yeah, absolutely. So it depends on the rate of division in the tissue that you're looking at. So if you're, and therefore the time. So for example, if you try and do okay, here's a good example. If you're doing Markham in the larval brain at L2, you get lots and lots of clones. If you're doing Markham in the L1 brain just 4 hours after it's hatched, you'll get only 1 of 4 neural stem cells dividing, because we know that at that time only those 4 stem cells are dividing. Everything else is quiescent. And if you do Markham now in the adult brain, you'll get nothing. Because I mean, right, just last year there was some evidence that there might, but until last year I would have said nothing dividing, because there's no adult neurogenesis in the flies. There's no cell division in the central brain of the fly in the adult. So it depends not only on your tissue, but on the tissue when your cells are dividing. Remember, Markham relies on cell division to occur. And the thing about toxicity, how the clone surviving will be a factor of your mutational analysis. So suppose you're using a mutant, you're making clones of a mutant that causes cell death. Then you may end up with a situation where the clones don't survive, or you'll see very small clones, like in the MYC mutant. Am I making sense?
Okay.
Anuradha?
Yeah.
Okay.
Sonia, what are these names? Those are
Don't worry about the names. That's the way they identify those projection neurons. They refer to the glomeruli, the part of the antenna. So this glomerulus, this region of the antennal lobe is called DM6. This region of the antenna is called VA1LM. So the neuron gets that name.
Yeah, and the numbers are the progeny. The number of cells, those numbers in brackets, 20 to 18.
Yeah, exactly. So it's telling you, so for example, this neuron was born when you gave heat shock anytime after this.
Correct.
Sorry?
Yeah, got it. Yeah.
And whereas DC2 was born only when you gave a heat shock in this time.
Yeah, and DC2 probably gave only 8 cells.
Yes, exactly. Yeah.
Sonia, I had a question.
Yes,
please go to the previous slide. Yeah, so let's say for neuroblast number 2 out here. So if you give heat shock during that period, so during the same time, the progeny of neuroblast 1, which is the GMC, which is dividing, wouldn't that also then get labeled along with neuroblast number 2? So it's like the progeny of neuroblast number 1 and the neuroblast number 2 are getting labeled at the same time. So,
yeah. So one clarification before I answer your question, Lamia, is that how you say your name? Yeah, okay. So this is actually one neuroblast. It's just a neuroblast that's dividing. So with each division, it's giving the GMC and 2 neurons. It's the same neuroblast, but it's just a little later in time point. Is that?
I get that.
Yeah. Now to answer your question, remember that it's still a random event. Even if cells are dividing and you have FRT, you have GAL4, you have GAL80, all of that, it doesn't mean that the recombination event will occur. Point number 1. Even if it occurs, it doesn't mean that independent assortment will result in a loss of the GAL80. So these are all chance events. So you are right. There is a possibility that when, suppose this cell lost GAL80, it's also possible that this lost. No, it's not possible with these two, but it's possible with, say, if this, let's see, if this, yeah, what is possible is this and this dividing at the same time. Remember these two are progeny of the same cell division. So if this has lost GAL80, this cannot have lost GAL80. This has lost GAL80, this has inherited GAL80, right? It's the product of the same division. Is that?
Yeah, and so it won't be for that neuroblast because I think the wave of neurogenesis will not allow the progeny of that neuroblast to divide at that time. Like, you know, when we have that.
Wait, just a second, Lamia. Let me just complete that thought. So in this neuroblast dividing, these are the two progeny. This cell loses GAL80, this cell inherits GAL80. So now everything downstream of this will be labeled. But while this is happening, you've given a heat shock, the flippase can still perdure. And so in this division, it can still lose GAL80 and one of them can be labeled. So it is possible that you do see cases where there's one neuroblast labeled and one event from a previous GMC along with it. But you will not be able to decipher those events.
Okay. So also then during embryonic neurogenesis, when there are multiple neuroblasts which are dividing at the same time, as in it occurs in waves. So in the first wave, let's say 10 of them are dividing. So again, will I be able, so again, then maybe a heat shock promoter will not, a heat shock promoter driven flippase will not be useful. You'd rather go with a neuroblast specific promoter. Because
you could do that. So Lamia, if you're thinking of doing that, I'll warn you that in the embryo, Markham has not been shown to work yet. So we don't quite understand why that is. But regarding your question, you know, even if you give a heat shock at L2, many neuroblasts are dividing. Still, there's a chance, only a certain amount of probability that you'll hit. So it depends on, you know, how accessible is the FRT, how long you give the heat shock. You can still titrate it to hit just a few neuroblasts. So at L2, almost every single neuroblast in the brain is dividing. Yet if you titrate your experiments depending on what FRTs you're using, how long you have the flip, you can induce clones in just a small number of them. That needs some fiddling. You have to understand your system. So you give a heat shock for some time, you'll see how many clones, okay, I need to scale it back a bit, maybe reduce the temperature a bit. So it's all tweakable.
Okay. I thought it was foolproof anyway. Okay. Now I get it.
No, that's the beauty of it. Because it is tweakable, you have the ability to label things randomly and sparsely so that you can see what's going on. Otherwise, you just use tubulin, galfo, US, GFP, and see everything, right? You don't want that. You want to restrict labeling to a small number so that you can actually decipher. See, the beauty of this is you can now see exactly how this one neuron is formed. But if you had this with everything else labeled, you wouldn't be able to see what that neuron is doing. So you want to restrict your expression patterns and randomly so that you can look at different things.
Sorry, just one last thing. And this is just to clarify. So once the GAL80 has been segregated and gone into one of these, or into the GMC and not the neuroblast, let's say, then from now on, whatever the GMC will divide into will always have that, right? So it's going to mark all its progeny at the end of it.
Yeah.
Thank you. Thanks so much.
Yeah, Dinesh?
One thing which is not clear, no matter how much you titrate it, right? B, C, and D are all going to be probable, right? So you will always.
But how many events of B, C, and D do you get?
What do you mean by that?
So for example, in the brain, there are 100 neuroblasts in each hemisphere. So 200 neuroblasts. So if you give a heat shock at L2, you don't want to hit all 100. So each neuroblast has the certain probability of giving you B, C, or D events. And you have 100 such neuroblasts there. So that was Lamya's question, basically.
Oh, sorry, I was linking back to the plot we had showed where they had figured out single neuron, so for them, they would have at least had to go through these 3 events and had to search through and find them. So they couldn't have avoided that, right?
No. So you have to dissect, and then pool your data and analyze it post hoc. Yeah, all chance. Yeah.
I see.
Okay, so let's just move ahead. So yeah, because we're already out of time. Okay, so basically you can combine it with anything you want. You can now put a UAS of a gene that you're interested in. You can put RNAi, and there are variants of twin spot, which are of Markham, which are twin spot Markham, which allows you to do the twin spot, but in a Markham setting, which makes it much more powerful, and dual Markham, which allows you to use 2 binary expression systems. So not just GalFO UAS, but Lexay operator as well. Okay, so I'm going to let you guys think about this again, since I've gone terribly slowly. Now that you've heard a little bit more, think about how you could use these tools in experimental biology for the questions that you're interested in. There are other ways in which people have used. So can I carry on for a little while longer, Deepti, and the class? What do you think? Sure, I'll be super swift and okay, it's 37. What do you guys think? Or I can give you the slides and you can just have a look at it.
Okay to go ahead. Yeah.
Okay.
Yeah,
okay.
You can go ahead. Yeah,
okay,
you can continue.
Okay. So the other way that people have used this is to trace developmental expression patterns of GalFO lines. So one problem if you work with GalFO lines is you look at it at a certain time point, but you don't know when it was expressed earlier. The GalFO lines are known for being very transient and temperamental. So this was G-Trace that was developed in Utpal Banerjee's lab, and it relies on, say, an enhancer GalFO that you're interested in, whatever tissue you're looking at, say the eye. And what it does is this method, it's called G-Trace, GalFO trace. You set up a cross with a fly that has the G-Trace, which basically consists of a UAS RFP, red fluorescent protein, but simultaneously also has a UAS flip. So remember we spoke of the UAS flip. So the enhancer GalFO, at whatever time you open up the brain, open up the animal, say L3, you will see RFP, which will tell you where the GalFO is expressed at that given time when you open up the brain. But because throughout development, it was also expressing flip, and in that genetic background is this construct over here. What this construct is basically a ubiquitous promoter that is driving a GFP, but that GFP is interrupted by a stop cassette like the CD2 that we discussed with FRT sites in tandem. Now, throughout development, if the GalFO came on in some other tissue or in some other part of the tissue you're interested in, then the flip would have resulted in an excision event, resulting in GFP expression. Now, because that GFP is being driven by a ubiquitous promoter, what you end up with is when you open up this animal, say you're looking at the wing disc, in red, you'll see where that GalFO is expressed now. And in green, you will see where that GalFO was expressed during development. So this is how they showed it in their paper. So here are 3 GalFO lines, hedgehog, patched, and DPP. And you can see that they're looking at L3 larval wing discs, and you can see in red, hedgehog is expressed in this pattern. And it looks like green coincides with that. So developmentally also it was expressed here. But look at patched over here. Patched, the expression domain during L3 is in this one nice line along here. But look at its historical expression. It was expressed in this entire compartment over here. And same true for DPP. It's expressed over here, but it's expressed also in this region developmentally earlier, which you wouldn't have picked up unless you used something like GTrace. So GTrace is really efficient if you want to, if you're working with a GAL4 line that you're not sure of what its expression pattern was in earlier developmental time point. So that's GTrace. Another very useful way in which clones have been used especially in the nervous system is to be able to label cells differently, because the nervous system consists of so many different types of neurons, and you want to know what, when you're trying to understand circuitry, you want to know who connects to what. And so you want to be able to label the cells differently to see where they're going, what they're doing, who they're connecting up to. So labeling cells differently has been done in very, very many different ways. So you basically, they're called the rainbow techniques, because you color each neuron with different colors. And there are very many different types. So there's something called flybow, which was inspired from brainbow, which was developed in vertebrates. Then there's something called bitbow. There's something called multicolor flip out, which is actually this image that you see over here. There's something called Rapley. There's something called tie and dye. And actually there are a bunch of others which I haven't, the coin flip, there's a bunch of them that people have developed as per their needs. So I'll just quickly go through a few of them to give you a feeling for how people play around with these things. But the purpose in this whole exercise is to label cells differently. So flybow relies on, again, flip FRT. So notice here that there's a UAS. So this would be driven by a GAL4 that you were interested in. There are two colors, fluorescent proteins, M cherry and cerulean. Notice that they are, that one is in the correct reading frame. You would have seen this type of orientation in Sonal's talk, I think, in her flip-flop cassettes. But in this way, whatever GAL4 came in, it would drive M cherry. And then there's this stop. SV40 is like a stop signal, so it won't drive anything beyond that. But because these two fluorescent proteins are flanked by FRT sites that are not in tandem but are opposing each other, what can result are inversion events. And we haven't spoken about inversion events in experimental clones yet. And so you can imagine that now if you came in with the GAL4 and you came in with the flipase, an inversion event could occur, so that now some cells may be labeled cerulean. So using this construct, you can get two colors at random, whichever orientation it happens in, whichever orientation occurs in a cell. So that's only two colors. You want, because there's the brain, you want many more colors. And so what these guys then did was the same cassette, UAS, with the same thing over here, but they inserted another exact same kind of cassette, but different colors, EGFE and citrine over here. And again, you have FRTs over here. So you can now imagine that when the flipase comes in, but now because you have four different FRTs, many different events can occur. So we discussed the inversion event over here. There could be an inversion event over here as well. But notice that this FRT is opposing this FRT. So you could have an inversion event over here, which would result in a different event. But notice now that this is in tandem with this. So you'd end up with a flip out event. And then this is in tandem with this, so you could end up with a flip out event. All of this to say that they've put all of this in. Now anything can happen, basically. And what you end up with is oh, I'm sorry, did I not put a picture? Ah, yeah. What you end up with is, when every time you open up the brain of these flies, they've crossed it to LV GAL4, the flybow, the second one that we discussed. Every time you open it up, a different event would have occurred. So you can see, it allows you to decipher things decipher patterns in an expression domain that you otherwise wouldn't have. And over here, there's actually one event which has occurred only in two cells, so you can decipher its pattern much better. So that's flybow. There's something called tie and dye, which actually wasn't used in the nervous system so much, but it's very beautiful. It again relies on a heat shock promoter. and 3 independent cassettes, each with a different ubiquitous promoter. Again, these cis FRT excision type of events, one driving nuclear GFP, one driving nuclear LAC-Z, and one driving GAL4. Now, excision events can occur, and if it occurs over here, then the GAL4 will drive RFP, but also a gene of interest. And what now you guys are familiar with this, you will end up with 3 events. Those independent events will be RFP, GFP, and LAC-Z, but the intersections of those will give you many different colors. So you end up with 8 colors because one of them would be no color. And so that results in these beautiful sort of images. And remember that the ones that are RFP are the cells in which the manipulation has also occurred. And a variant of tie and dye is Rapley. And Rapley was developed, it's almost exactly the same principle, except that these labels are fluorescent proteins, so you can image them live. And that was the huge advantage of Rapley over this. But they were also looking at wing discs. MCFO, I showed you a picture of, but it basically relies on something called the spaghetti monster GFP, which is this ball of wool over here. It's basically mutated so much, the GFP, that it's not fluorescent, but you can put many tags onto it. And those tags, there are many, many, many tags. And not only that, they've put a myristylation signal. That means that it'll go to the membrane. The disadvantage of tie and dye and Rapley are that because in neurons you want to see these long membranes, tie and dye and Rapley weren't very useful for neurons, but MCFO is, because they've got this myristylation signal. And again, it's the same idea that that independent excision events result in individual colors, but combinations will give you different colors. And so what would have been a GAL4 expression pattern, which is broad, again, this is downstream of a UAS. So whatever if you're interested in a particular GAL4 expression pattern, which if you just crossed a GFP would be a mess like this. If you have a small number of clones, you can see individual neurons and their morphologies. But if you have lots of clones, this was the idea, Anuradha, of longer heat shocks, shorter heat shocks, then you end up with lots and lots of clones. So they look pretty, but it's hard to decipher what each neuron is doing. Okay, and so that was labeling cells. And then a final thing, which actually I'm not going to talk about, you guys can breathe a sigh of relief. And the reason I'm not talking about it is because I think Sonal would have covered it in her talk, which is to be able, see, all of what we've discussed so far, except for the MCFO and things like that, where you have cis flip events, rely on cell division. But to probe gene function in post-mitotic cells, that doesn't really work. And so actually, I thought that Sonal was going to talk next. But basically, she's developed these tools that are based on the mimic lines using flips and ATP sites that allow you to flip orientations and visualize cells either in a protein trap sort of way, where the protein is labeled with GFP, but you cause an inversion event that creates a stop cassette, but also puts an RFP. So you can probe gene functions in post-mitotic cells using setups like hers. So that's another way of using clones. Okay, so the bottom line, I'm ending. I'm very sorry for going over time. The bottom line is that clones are terribly powerful tools in genetics. They're extremely versatile. You can use them. You can really fiddle around with them and orient them put them in configurations that would get you to get them to do the thing that you want them to do for you. And so you really need to think about the experiments that you want to do and use all of these bits and pieces, combinations of GAL4 lines, GAL8Ts, different Q system, let's say, and combine it to answer the question that you want. So bottom line is be creative with clones and have fun with them, because really, I mean, you saw those images, they're really nice to see. Okay, and I will end with if any of you have any questions. I would be happy to answer them.
Sonia, I had just one question.
Yeah.
I think I might have missed that. So can you just go over the significance of having the FRT sites close to the centromere?
That was done by design, because, you know, if you put it close to the centromere, then any gene that anyone is interested in can be recombined onto it. If you put it somewhere in the middle, then the gene before that, a recombination event will not result in a homozygous. Is that making sense?
Does it make sense? Because then the centromere, since it's spanning the middle of the chromosome, so it can involve genes on either side.
No. So if I draw here, can you see? Can you?
Yeah, I can see it.
Yeah. Okay. So
you can draw on the top as well. It's fine.
Over here, can I draw here?
Yeah.
Okay. So here's a chromosome. Imagine it's the second chromosome. Okay. So what the stocks that are available are stocks with an FRT here, and it's placed over here so that any gene downstream of that on this arm will give you the chance of getting a homozygous event. In a different stock, people have made FRT sites here, so that now any recombination event with a homolog here will result in any one of these getting swapped. The recombination event will occur right here, so that the entire chromatid after that can be swapped, and so you can end up with a homozygous null. If you had it somewhere here, and so if they made a stock that with an FRT site over here, if I had a gene that was here, a recombination event would never result in a homozygous here, because the recombination has occurred downstream of the gene I'm interested in.
It's clear now.
Yeah, okay. Another thing that I didn't actually discuss was people have used FRTs slightly off. So if you have these two, if you have an FRT site here and an FRT site here, by combining them, you can actually create deletions. But that doesn't really fall under the realm of this. But there are stocks that have FRT sites at different locations in the chromosomes. Okay.
Are there any potential drawbacks to this? Something
like what, Dinesh? Like, what do you mean by drawbacks?
Things to be wary of, things which might give rise to false interpretations.
It would really depend on the way your experiment was set up, Dinesh. So for example, when you're working with GAL80, remember that you can always get rid of GAL80, but you can't induce GAL80, so you can't shut it off at some time. So that's an issue. Sometimes you can't really tell exactly when things are born, because you imagine a cell division scenario where a recombination, so remember the recombination is happening at interphase basically, right? It's not happening during mitosis. So imagine a cell does all of that business and then sits around for 2 days for mitosis to occur. That happens. It's not unusual. When cells go into quiescence, they go into whatever G2 arrest and things like that. And then they come out of it, 2 days later, and then they divide. So you would think that this cell is born 2 days later, but actually it was born at the recombination event occurred. You would think that it was born at an earlier time point, but actually the mitosis occurred 2 days later. Is that?
I see.
Yeah. That's one in birth dating that would be an issue. Markham hasn't been shown to work in embryos. I mentioned that. That's a drawback. This business of titrating, how many, what's the number of cells. That can sometimes, if you're working in a tissue that's completely new, you don't know when the cells are dividing, that's often an issue. With the multicolor labeling, again, unless you get the density right, it's very hard to say. So for example, that last image that I showed you, it's beautiful, but it's hard to say anything happening. So combining those with mutant analyses are very, very hard. Yeah, I mean, there are advantages and disadvantages to a whole bunch of things.
So are there better approaches to get these probabilistic sort of neuron labeling than, you know, titrating temperature? Or,
yeah, so one way of doing that is to fiddle around with FRTs. So yeah, I spoke of the FRTs as if they were one thing, but actually there are different types of FRTs. So in the palindromes, there'll be some changes in nucleotides, which makes them exclusive. So for example, there'll be FRT 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, depending on those few differences. And within those FRTs, you have to have a homologous FRT for a recombination event to occur. So what people do is they say, okay, I'm sick and tired of this. There are 100 neuroblasts. Every time the smallest heat shock I give, I get 10 clones. That's not enough. So then what they'll do is they'll try and either come in with a less efficient FRT or combine FRTs such that you have some dummy recombination events. So you create dummy recombination events that won't result in a labeling, but those dummy events preclude a real event occurring. So that allows you to titrate it further. But you really have to work with your system to see what's the ground reality, and then decide how to titrate. Is just shifting temperature enough, or do you need to go back and re-engineer FRTs and make new stocks?
I see.
Okay, I think.
Are there any more questions?
And if something strikes you, don't hesitate to write to me. I should have put my email address there, but it's Sonia, S for Sen, SONIA @instem.res.in, or it's the other one is very easy. It's sonia.sen @tigs.res.in. So yeah, feel free to shoot me an email and I'll be happy to help with anything.
Thank you, Sonia. So I have a announcement to make, and then we will stop the session. Just a minute. Sorry, there was some sound. Yeah, so Aman is asking for tomorrow's session whether you would prefer a presentation-based talk or hypothesis building behavior experiments. This is something you can write on chat, 1 for the presentation and 2 for building hypothesis. You know, then I will let him know what people.
That's very mysterious. What does building hypothesis mean?
It is mysteriously written to me, so I have no idea. And I think I'm just going to tell him that hypothesis building experiment, because this is already, I haven't seen anyone yet. But if there is anything, I mean, anybody who prefers presentation based, do let me know. Otherwise, I'm going to go with 2. Okay, great. So thank you, Sonia. It was very useful. And these images are so beautiful. I mean, I'm thinking that you are also trying something similar with mosquitoes. You're going to make these constructs for mosquitoes as well, right?
So that's, yeah, exactly. So we're trying to make some neuroblast clones in mosquitoes. And again, you know, we're doing exactly, we have no idea where are the neuroblasts, are they dividing, when are they dividing? So we're basically designing a few constructs that are doing this kind of titrating, Dinesh, where we're, you know, some we're keeping a standard FRT, some we're combining FRTs. And so yeah, we'll see where it goes. Maybe next time I do this, I'll have some pictures from mosquito brains to show you.
Yeah, I'm really looking forward to those to seeing those experiments. Yeah.
Okay, thank you all very much. Good luck and enjoy fly pushing.
Thank you so much.